Hello, everyone. Welcome to the seventh CAPS Talk series, where we're talking about protein structure this month. So last time we heard about cool path toward protein-protein commitments. Today, we will be hearing about dialing into structural domains. Over to you, ma'am.
Yeah, thank you. Thank you, Shaila. All right. So you might recall that we had talked about domains when we were in the sequence team, and we had appreciated at that time that it is possible to have different domains of different complexity. And this one is an example of a homeodomain where there are two distinct compact regions which are easy to recognize. There is, for instance, a kind of domain which is in the blue and green, and then the entire domain is shown in orange and red. It follows it. It seems to be a very simple domain architecture. We had discussed that. And we had also discussed subsequently that there are other complicated domains. For instance, we can have such an arrangement as shown in this E-structural protein, where we can have certain three proteins that come together and form one domain that is shown as 3 helices, and then the rest of it are contained as other domains. And here it seems to be almost like they are girders, and they are holding the larger domains as it were. And this can mean it becomes so important to appreciate the presence of multiple domains that constitute a protein, right? And we had discussed this in the context of the sequence theme, just to show that could be quite hard to recognize them. And it becomes even more hard in this example, as shown in this example, which is a hypothetical protein where two domains are so intricately interacting, and the chain also goes back and forth to the two domains, and it becomes so hard to recognize the connections. However, in the context of the structural theme, you can see how it also is becoming hard to recognize the domains. And years ago, and much before even the lab had started in 98, as a part of my postdoctoral work, I had initiated some kind of objective algorithm to recognize domains. And why algorithms are required to recognize domains, we are able to see so easily and not so easily in some cases. And the reason is simple. Because as long as there are a handful of proteins, we can just happily would have been looking at domains by means of looking at the graphics and writing down where are the domains starting and where are they ending. However, even in the protein structural data bank, so many entries come in day by day, and the numbers are growing with time. And therefore, objective algorithms become important simply to scale up. And the other is the objective algorithms have advantage because what I say as domain boundary could be different from what another person says. And even what I say before lunch or after lunch could be different. So therefore, objective methods are important. And this is a last example that I want to bring up of a lactate dehydrogenase. Here, there is absolutely no problem in recognizing the domains as compact regions. You can see one domain to the left, one domain to the right. However, in terms of the structural excursions, you can see that the N-terminal region and the C-terminal region together forms one domain to the left. And the second domain seems to be constituting middle region of the protein. So I had theorized that why not concentrate on the secondary structural elements, namely the helices and the beta strands, and examine the inter-secondary structural distances, wherein we take, for instance, one helix and then look at the distance that helix subtends with the neighboring helix. And in fact, we will do this for all possible pairs of secondary structures. So we are left with a matrix of distances, average distances, and then we can, why not put it through the classical phylogeny techniques as shown here. So each of these elements turn out to be secondary structures. It could be H 56 to 69, meaning residues starting from the 56 to 69 form one helix. It seems to be spatially close to a beta strand, which is from residue 23 to 27. So then all of these come together to form a cluster, and this is nothing but a spatial cluster. And such objective methods could be useful then to recognize structural domains. Wherever there is a cluster of secondary structures in the dendrogram, it could completely then translate to a structural domain. And this method was called as dial. And as I said, this was formed much before the lab itself was formed. It is pre-98, maybe 94. I invite Meenakshi to present how these sort of dial methods then could have been organized as a form of a database. And this is called as DDBase. So may I request Meenakshi to come online and present the DDBase part.
Thank you, ma'am, for the introduction. So I'll just give a brief overview about the domain identification algorithm, which is DIAL, and the application of DIAL on the entire structural repertoire, which is the PDB database, in the form of DIAL-derived domain database, which is DDBase. So these are the links to access the DDBase and the DIAL database from the lab. So I'll just give a brief overview of the DIAL algorithm. So the DIAL algorithm consists of it is an amalgamation of multiple algorithms which perform different calculations. So one of the calculation is to look at the proximity of the secondary structural elements within the PDB structure. Supposing we have a PDB structure, how many secondary structures are there, and how much distance or how close to each other these secondary structures are there. So we use the algorithm called as struct, which reads the PDB file, calculates torsion and the secondary structure assignments. And we also define something called as a proximity index, which is the extent to which different pairs of secondary structures interact with each other in a protein. And then we calculate average of all the possible distances between the C alpha atoms of one secondary structure to the other secondary structure using this formula, wherein we say that Ni is the number of residues in the secondary structure I, Nj is the number of residues in the secondary structure that we are comparing, which is J, and dKl is the distance between C alpha atom of kth residue in secondary structure I to the C alpha atom of the lth residue of the secondary structure J. So using this proximity index calculation, we identify the distance of all the C alpha atoms between different secondary structures. Following which, we use another algorithm called as the Kish algorithm. And this algorithm will cluster all the segments of secondary structures based on the proximity indices, which is basically based on the distances between the C alpha atoms in one secondary structure to the other secondary structure. Then following the use of the Kish algorithm, we use the disjoint algorithm. And this disjoint algorithm will identify clusters from the output of the Kish program. So these clusters will then be represented in the form of a dendrogram. And these using these dendrograms, we can identify the different the clustered regions within the PDB structure, and these clusters later on, using another calculation called as disjoint factor, we can identify if these clusters are indeed distinct domains within the PDB structure. So we will next look into what is the disjoint factor. So let us consider a situation where n clusters are identified from using the Kish and the secondary structure dendrogram of the protein. So the disjoint factor will give the extent of physical interaction between the identified clusters. So if there are, say, n clusters identified within a PDB structure, the disjoint factor will let us know what is the extent of interaction between each of these group of secondary structures. And it is calculated using this formula. And here in this case, alpha is a ratio which is an expression of the total number of secondary structures within the protein, the proximity index calculations between the secondary structures, the number of secondary structures within each color clusters, and the weight attached to each pair of clusters. So after this disjoint factor calculation, so for each possible scenario, we'll get multiple disjoint factors. So for this, to identify the domains, there can be multiple possible solutions based on the interaction of the secondary structures with each other. So, and for each possible case, we look at the disjoint factor, and then we take the best possible solution as that solution which has the highest disjoint factor. And then we say that, so supposing for this particular PDB structure, there are multiple solutions and the best disjoint factor is a disjoint factor of 1.2. So we take that domain description, and in that case, if there are 3 domains, we have identified 3 domains, 3 structural domains within the PDB. And so the dial algorithm, as Sowdhamini ma'am had mentioned, she had worked on it in '94, the original version of dial. And after this, in the lab, there have been several iterations of dial, several versions of dial. And in each of these cases, we start with the secondary structures, and then from dial 1 onwards, even loop regions are taken as secondary structures for the clustering calculation. And in the later versions of dial, like currently we are using dial version 4. So we are splitting longer secondary structures such as helices, strands, and loop, if they are longer than say 15, 10, and 9 residue segments respectively. And we are then doing the clustering calculation to assign the disjoint factor to each of these secondary structural distances. So like once we have this dial algorithm, and supposing as from a user's perspective, if we would like to use the dial algorithm or the DD base database, so we need to know how to interpret these disjoint factor values for the solutions. So we I'll be showing a few examples here. So if the disjoint factor is less than 1 for the entire PDB structure, then it means that there is a single domain in the protein, in the PDB structure. But in case of the value slightly higher than 1, which is between 1.0 to 1.15, this is either slightly borderline cases. Either there is a single domain, or there are very, as Sowdhamini ma'am had mentioned, that in some domains they are very intricately interacting with each other. So here we may have such cases of conjoint domains. So within the values 1.16 to 1.25, we are calling those structures as conjoint domains, where there are two domains, but they are very their secondary structures are very much interacting with each other. So here we have the example of rat masked cell protease 2. So here we see that there are secondary structures between 2 different domains which are extensively interacting with each other. Then the next scenario are the interacting domains. So slightly higher disjoint factor values between 1.25 to 1.5 are the interacting domains. And in this case, we can see the example of para-hydroxybenzoate hydroxylase, where we are seeing that there are some secondary structures which are interacting from one domain, which are interacting with certain secondary structures of the other domain. And in the third example of phosphofructokinase here in the disjoint domain, we see that there are, even visually we can identify that there are 2 distinct domains in the protein. So in such cases, the disjoint factor values are usually above 1.5. And so with this, I will switch gears and talk about a database wherein the entire structures, the salt crystal structures in the PDB data bank have, in that database, we have tried out the dial algorithm, and we have the results we have populated in the form of a database. So in the DDBase database, we can navigate the database by looking at, by going to the entries tab. And in the entries tab, so all the structures in PDB will be listed out, and we can click on each structure to get into the results. That is how many domains are there in each protein, and how many of them are interacting with each other. Are they distinct domains or are they single domains? We'll get an idea about the domains within each PDBI. So here I would be showing an example of the PDB ID 119L, which is a lysozyme structure. So this, so once we click on this entry, we get the results. The result is by default the best domain definition. So best domain definition is the among all possible solution, the one which has a higher disjoint factor, highest disjoint factor. That is the best domain definition. So here we are seeing that there are two distinct domains. The disjoint factor is 1.2, and we can see that the first domain is consists of only alpha helices, so it says here all alpha. The second domain has alpha helices plus beta strands, so it says here all beta, and these are the number of residues, 81 and 73 respectively. And then we see that the residues of 75 to 155, 3 to 75 have been assigned to these two domain definitions. So these details can be obtained by clicking on the best domain definition. Then if we would like to see what are the different sequences, the secondary structures, and view the structure in RASMOL, so we can see that using the sequences secondary structure RASMOL tab. And here I have shown a description, a cartoon representation of the structure, where you can see this is the lysozyme, the PDB structure 119L. This is a T4 viral lysozyme structure. And here you will get the sequence detail. This is a lysozyme enzyme which belongs to the hydrolase category of enzymes. And we'll also get the sequence, the amino acid sequence of this entry in the PIR format, directly derived from PDB. And then there are several other results which are calculated in DTBase, which we'll be seeing now. So we also get a distance matrix of all the secondary structures. So there is an all versus all distance calculation between the secondary structures, which is done at the Kish algorithm stage. And here we can see that for the first helix, which is between residues 3 to 11, you can see that these are the distances with the other secondary structures. So all the secondary structures are being listed below. So I'm just showing the starting few secondary structures. So here H is for the helix, E is for an extended beta strand, which is between 14 to 19 residues, and then there is a loop of between 20 to 24 residues. So the next result that we get is a rough dendrogram, which has been calculated using these distance matrix. So this shows which set of secondary structures are closely related to each other based on the distance calculations. Then you get a cluster picture in the database, so using which we can again see which set of secondary structures are closely clustering with each other. Then we get a table of other possible domain definitions using which have been calculated from the dial algorithm. So here we can see that among all the possible domain definitions, the second situation, which has a disjoint factor of 1.2, which was the best solution that we had identified, that is the best domain definition. And here that is between node number 17 and 1. So here, when we look at the rough dendrogram and the cluster, we can identify which are the clusters, the node numbers we can identify, and then we'll be able to correlate between using the result table. And so these are all the results that we get, and then we'll go on to how to interpret these results. So supposing there's a disjoint factor of 1.2 and there are two domains identified, how to interpret this. But before that, I would like to say that we can also query the database using protein sequences by using the BLAST program, which has been implemented against all the domains for whom we have the domains we have calculated for the PDB entry. So again, using the protein FASTA sequences, this database can be searched. And once the search is done, the results of all the sequences in the database, which are closely related to the sequence that has been queried, will be displayed. And again, similar type of results can be obtained for those sequences. the output which we get from the database. Then, going back to the previous example of T4, the phage lysozyme, we see that two domain definitions were proposed by Dial. So here between residues 3 to 75 and 75 to 155. So these are structural domains which have been proposed. And for the same PDB, which is the ID is 119L. So we see that PFAM, which is a sequence database, is proposing a single domain, and even other structural databases like CAT, SCOPy and SCOP2B, they are proposing a single domain definition. But upon literature survey, we can realize that, and even in our case, we have identified two interacting domains. So beyond 1.5 disjoint factor value, we can help, that will help us to distinguish between the domains. So in our case, this is within the 1.5 value. So these are closely interacting domains, interspersed domains. And in this case, we are also seeing that based on the literature survey, we observe that these domains are easily distinguishable but energetically coupled. So in during the folding state studies, I'm sorry for the technical issues on my side. So I've logged in again. So I'll get back to the domain definition. So compared to other databases, we have identified two different interacting domains using Dial. And here we see that these are very closely interspersed interacting domains, which is seen by the value of disjoint factor. It is less than 1.26. So there are no two distinct domains, but these are closely interacting. And again, upon literature survey, we realized that these two domains are also formed during the folding stage. So structural domains by definition are individual folding units within the protein. So during the folding stage of lysozyme, the C-terminal domain gets folded first, for the C-terminal region gets folded first, followed by the N-terminal domain. So again, we can relate structurally that in our case, we have obtained two different domain definitions for lysozyme. Thank you, ma'am.
All right. Thank you, Meenakshi, for a very clear presentation and bringing up the examples. And next, I will like to explain to you how these sort of finding domains using Dial made us land up on a very interesting observation, which is that of the domain swapping. Right? As we were looking at the Dial results, there were multiple examples where more than one chain together were forming domains. If you have a multimer, multiple chains were together forming one domain. And that led us to go back into the literature and find out what they are. Because multiple chains coming together and forming multimers is all right because it's a quaternary structure. However, why is it that multiple chains together form one domain? So in that context, we landed upon this very interesting phenomenon, which is the domain swapping. And then when we looked in the literature, in Professor Tom Blundell's group, they had already reported the gamma crystalline, whereas it forms two domains neatly like this, as shown on top as A. Beta crystallines, beta B2 crystallines form domain swapping. And in the case of the diphtheria toxin from David Eisenberg's group, they had actually reported multiple chains that are swapping one domain from one chain to the other. And they had also coined the term swapping. And what is this swapping? We can imagine that in the open form, this particular protein, diphtheria toxin, might open up the loop region that is going to present its domain, the rest of the domain onto the other chain, right? So normally it is closed like this, And then some point of time, there is a conformational change of this loop region. And therefore, this particular domain then kind of goes and latches onto the other chain. And it happens the other way around as well. So it is a symmetric sort of giving part of its chain, the domain, to the other. And this is quite interesting. But how can we follow it up? And why is it important? Because domain swapping could vitally be giving rise to unlimited such interactions. Instead of just two chains coming together, imagine if it happens many, many times, then it can give rise to aggregation. And aggregation, protein aggregation, definitely has implications in neurodegenerative diseases. So therefore, we got interested. And I'm going to explain some of the terms so that you can appreciate the structural analysis that emerge from the domain swapping. And this little circle that has gone from one to the other chain is called as a swap domain, right? However, that particular loop that had undergone the conformational change is referred as the hinge. The protein might have many, many loop regions, but this is the loop that is changing its conformation so that this little domain can go and sit on the other chain. So that is called as a hinge. And the primary interface is something what the two dimers, the two protomers are already engaged in. However, there is a newly formed interface that arises between the little domain that has gone to the other chain, right? So those are referred as the swap domain interface, which is that little region, and non-swap domain interface, which is the part of the protein interface, which was always there. It was always present between the two chains, and that is called as a non-swap domain interface. So I might be using DSI to refer to this little part, and the non-swapped interface is called as NDI. All right. So with this definition being introduced to you, we will be playing the recording of one of the students, Prashant Shingate, who had done a lot of the structural analysis of domain swapping, and consequently our appreciation of amino acid compositions, especially at the hinge region. All right. So Prashant's recording is going to be played just now.
Hello, I'm Prashant Shingate. I was PhD student in Professor Sowdhamini's lab from 2009 to 2015. Presently, I am R&D scientist at Bayer, Singapore. I would like to congratulate ma'am and whole CAPS team for completing 25 years milestone. I'm glad that I was also part of it. Today, I'm going to share a part of my PhD work on domain swapping. I trust Professor Sowdhamini have already explained the domain swapping concept. However, I would like to reiterate some terms which I'll be using throughout this presentation. 3D domain swapping is an oligomerization mechanism in which proteins form intertwined protein complex. In domain swap dimers, there are swap domains which moves from one monomer to another. Then there is a small loop-like structure known as hinge region which allows movement of swap domains. Before oligomerization step, here monomeric proteins partially unfold and form open conformers. These open conformers intertwine and form domain swap structures. In domain swapped oligomers, there are two types of interfaces. An interface formed by swap domain is known as swap domain interface and will be referred as DSI in this presentation. Then there is a newly formed interface which does not involve swap domains, hence it's called as non-swap domain interface and will be referred as NSI in this presentation. The most interesting part of domain swapping is the hinge region. The hinge region is the only region which changes its conformation during domain swapping. We found that these hinges adopt C-shaped structures in monomeric form, whereas they adopt extended structures in oligomeric forms. As you might have imagined, the hinge in C-shaped loop appears more strained compared to the relaxed extended form. These are phi-psi angles of hinge residues. Those in yellow are hinge residues from oligomer occupying more stable regions in the plot. The hinge residues in monomeric form shown in red color, and they adopt more strained position than oligomeric hinge. This strain in monomeric hinge conformation might be one of the driving force for forming partially unfolded open conformers and ultimately favoring the domain swapping process. Hinge regions are mostly loops. Then why certain loops act as hinge and not rest? Hence we decided to check amino acid propensities for both hinge and loops. We observed certain amino acids like alanine are more frequently present in hinge compared to loops, whereas some amino acids like aspartate are more prevalent in loops than hinge regions. The green arrows here represent the experimental evidences, which shows substitution of amino acids from the bottom of this list with those in the top of this list can induce domain swapping, whereas the red arrows denote vice versa, can inhibit the domain swapping process. So this order of amino acids can serve as a reference to control the domain swapping process. After hinge regions, analyzed interface regions. As mentioned earlier, the domain swapped interface needs to be denatured to form open conformers, which in turn is critical to form domain swapped oligomers. The energy required to partially unfold monomer decides the height of the energy barrier while transitioning from monomer to oligomer. Hence, we decided to study what kind of interactions are present in DSI and NSI. We observed on an average, DSI contains much more interactions per unit area than NSI, which suggests that there are more optimized and stable. The DSI are present in both monomeric form and oligomeric forms, so they get more chance to evolutionarily optimize, whereas NSI are newly formed interfaces, hence they are not well optimized. Next, our amino acid propensity study of interface regions revealed the presence of more charge residues at DSI than NSI. There are several studies which suggest by changing pH we can induce or inhibit domain swapping. We think the presence of several charged amino acids at DSI might be the potential reason for this. Hence, we decided to study this further and collaborate with Professor Jim Waviker from Manchester University. Professor Waviker developed a pH dependence program which simulates changing pH conditions and then calculates the energy of interaction between two subunits. We applied this program on different domain swap proteins for which we have both monomeric as well as domain swapped crystal structures available. This is one of the example. This graph shows a pH-dependent energy profile of DSI and NSI of diphtherotoxin. The DSI profile is shown in red color, while NSI profile is shown in blue color. This protein is monomer at pH 7.5, whereas it forms a domain swap dimer at pH 3. We observe destabilization of DSI at pH 3, whereas stabilization of NSI at pH 3. In other words, uh, we observed weakening of DSI and strengthening of NSI at pH at acidic pH, which favored formation of domain swap structures. Let's see this in detail. After observing the structure of diphtherotoxin dimer, we found three salt bridges present at DSI, which were destabilized at acidic pH. Whereas we observed four negatively charged amino acids with repulsive interactions at NSI, which became neutral at acidic pH, which caused the increase in stability of NSI at acidic pH. Thus, weakening of DSI and strengthening of NSI lead to formation of domain swap structures at acidic pH. Similarly, we observed several other domain swapped proteins and we found weakening of DSI and strengthening of NSI were one of the major reasons for pH dependent domain swapping. To summarize this talk, the strain in hinge region, weakening of DSI and strengthening of NSI are major forces for domain swapping process. The amino acid propensity studies were nicely correlated with the experimental evidences, and we strongly believe that it can provide further hints that which mutations can induce or inhibit the domain swapping process. So that's all I have for today. I would like to thank Professor Sowdhamini for the guidance and support. My collaborators, Professor Jim Wavika, Dr. Atul Upadhyay, and Dr. Shamil Khadar for their contributions. These are the publications related to this talk. Please feel free to explore them for more details. Once again, congratulations on this 25 years milestone, and thank you for your.
That's great. Yeah. So I think you'd also be able to appreciate that there are lots of important influences by the environment on the whole phenomenon of domain swapping. The pH is one of the major influences, and we have been able to collaborate with Jim Waviker, and Prashant had very nicely explained that. All right, so we'll go back to our discussions on what more one can do. And I had mentioned that we can apply this in the context of neurodegenerative diseases. So can we predict say, the human genome, how many proteins are likely to be engaged in swapping? So that's the next question we were asking. And since we have a rich data set of domain swapped examples, can we use that for the prediction? And here, as I said, historically, we bumped on this phenomenon of domain swapping purely from our application of dial and on large PDB entries, as nicely explained by Meenakshi as well. So when we were doing this uplift of dial, applying dial on all the PDB entries and creating the domain database, there were appreciable number of examples, as I said, that multiple chains, more than one chain together forms one domain, as opposed to the cases where one domain is from A chain and another domain is from B chain. You will find that both A and B constitute a domain. And that's how one can begin to accumulate possible domain swapped entries. And now we are creating a database for domain swapped entries, right? And so dial definitely is the first step in accumulating possible domain swapped entries. And then we applied some filters so that we are not looking at some small loop regions transiting from one chain to the other, or it may be some kind of other spurious example. So we applied a symmetry check because whatever is being lent to one chain, the same is being given by the other chain as well. So we applied the symmetry check, and also we had consulted the literature to see what the crystallographers were saying when they were reporting such structures. And altogether, this gave rise to what is called as a positive data set. And all our runs and our accepted entries as 3D, the domain swapped protein examples, protein structural examples are contained in this data, are contained in a database that we created, which is called as 3D swap. And it's in the public domain, it can be accessed. And in fact, we have also made further updates to this database. And in the context of the prediction, however, this will form what is called as a positive data set, because these are definitely examples which where even from structural information, such proteins have been engaged in domain swapping, right? And we were going to apply machine learning algorithms. And for machine learning algorithms, you need a positive data set and you need a negative data set, and you need a whole lot of properties that go into the training, so that we can begin to understand what sort of properties are crucially influencing or determining whether a protein is likely to be engaged in domain swapping or not. So what is our negative data set? That is a very tricky one because we never know those proteins which are currently not reported to be engaged in domain swapping will never ever be engaged in swapping. However, we created a negative data set so that they will never fall into any of these folds. So that was the data set. And in the properties, to begin with, we performed some structural analysis that Prashant also alluded to. Professor Janet Thornton had shown years ago that the domain, when two proteins interact, at the interface, it is much more planar. On the other hand, you can see that in the domain swapped entries, there is an appreciable amount of protrusion going on, a convex nature. So that part of the newly domain swapped interface is not planar, but engaged in a structural indentation or a protrusion. Likewise, the van der Waals interactions are much more at the domain swapped interface in comparison to the regular non-domain swapped interface. And the hydrogen bonds also, there is a mild preference. So already this was suggesting to us that indeed the domain swapped interface has some structural properties that could hopefully help us in a predictive scale, because in machine learning algorithms, these all could go in as our properties. So geared with the positive data set and the negative data set, we were going to use a random forest method to train, to get to train. And these were all the properties, and sometimes you use combination of these properties, and these are all at the amino acid level. So after this, we are not using structural information at all. And then we used techniques or properties such as isoelectric point, percentage buried residues, and so on. And together, we perform what is called as a training. And for this, as I said, we use random forest, where each of these properties are used to influence and provide a vote as to how well the member in the positive data set is kind of accepted in that way, and how different combinations of properties can also go into the voting scheme. And the system gets trained. And since our positive data set had about 2,000 entries, if the training had been 100% perfect, we would have got 2,000 true positives and 2,000 true negatives. On the other hand, the method was not able to recognize all of them. And there were indeed 237. False positives means they are been identified as involved in domain swapping, but it is not truly the case. It actually belongs to the negative data set. So and there are a whole lot of statistical parameters such as accuracy, specificity, sensitivity, and Matthews coefficient, which are usually done. And these are measures to tell how well the training has gone in. And accuracy such as 86% is considered to be quite good. And then we used our updated 3D swap database and the newer entries to then use it for testing. And in the testing also, we had about 216 members in the positive data set and 216 members in the negative data set. And again, if the testing is 100% efficient, we would have got 216 true positives and 216 true negatives. But the good news is that the accuracy is not too bad. It is again 84%. And since we were getting good results, not only in the training data set, but also in the test data set, we went on to applying this at the whole genome. And this is the part that Atul is going to present. All right. So I would request Atul, who has come online. He will be presenting how this prediction method could be was applied at the whole genome level, not only in the human genome, but few others. Atul, please.
Thank you, ma'am. Thank you. First, let me share my slide. Good afternoon, everyone. I hope I'm audible to all of you. If someone confirm if I'm audible.
Yes. Yeah, we are able to hear you, Atul.
Okay, thank you, ma'am. Right. So as ma'am has explained the method, how we developed these prediction models, and then as she showed confidence on me that I was interested more in the genomics at that time, and she asked me to use these models in the genomic level. So that's the title of my presentation today. So as ma'am introduced, I am Atul Upadhyay, working as assistant professor in the Department of Biotechnology, Thapar Institute of Engineering and Technology, Patiala. It's in Punjab. Right. So my this part in this part I'm going to discuss about the application of three-dimensional domain swapping prediction models to genomes and detailed annotation of the predicted proteins. That means, let's say we are going to find that these are the protein entries which will be involved in domain swapping. So what are their annotations, like what kind of protein families they are, or what are their function? Such is there any type of enrichment in case of diseases and all that we are more interested, and we try to say in this case. So starting with the let me pull this down. Yeah, right. So first we started with the prediction. We used our prediction model on the on different genomes, but main emphasis was on humans. And then in the second part of my talk, I will be discussing about the application of these models to some plant genomes, where we were interested to see what kind of proteins are involved there. So we started with something like we take the proteomes, mainly the reviewed entries from the UniProt, for the prediction of the three-dimensional domain swapping. And we have selected many of these genomes, and I'm going to emphasize only in case of the humans, what are the type of proteins involved in the domain swapping, right? So basically at that time there was around 20,000 proteins present in the human proteome which were reviewed. So we took all these proteins, and by using all these methods, models which we have developed, mostly the random forest one where was applied to predict the domain swapping, and we got around 11,500 protein sequences which were involved in the domain swapping, and which is around 57%. Likewise, we have applied in the different genomes, and similar results were obtained in case of domain swap cases. Right. So once we got the domain swap cases, like these are the, let's say 57% of the proteins which were involved in domain swapping as a prediction with the accuracy of around 85%. We want to see what kind of these protein sequences are in terms of what domains they are, or what is the domain architecture present in them, so that we can have an idea about the functional aspect of these proteins. And then we found that most of the domain swap cases were in these families, mostly in protein kinase domain or immunoglobulin-like proteins. With there were many other domains present, but most of the enriched domains were these ones. And these domains, when we checked their domain architecture, they were around 2,346 different domain architectures. And we all are familiar that these domain architectures give you the different functions. That means change in the architecture will give you a different function. So that's why we were also interested to see the domain architecture of these proteins. Similarly, we looked the enrichment of these domain swap cases in different metabolic pathways, and then we found that protein ubiquitination or protein glycolation pathways are highly enriched in domain swap cases, whereas there are many more metabolic pathways where these proteins were involved. So totally we reported that there were 254 metabolic pathways where these domain swap cases were present. Likewise, we looked the gene ontology of the domain swap predicted cases, and then we found that there are certain ontology groups or processes which are highly enriched in case of the domain swap predicted entries, such as in the organelle genomes or in the binding activity or maybe cellular process. All these were the enriched cases in case of these domains. We also try to find out the presence of these domains predicted cases in different protein families, and then again few protein families were highly enriched for domains predicted cases. So to summarize this part, almost 57% of the protein sequence of human genome were predicted as putatively involved in domain swapping, and the similar trend was also observed in across genomes. Then we finally did a detailed enrichment analysis in terms of protein domain families, gene ontology, pathway analysis, and their involvement in diseases, and which revealed that the functional classes of the proteins involved in domain swapping. Yeah. Side by side, we also looked at KEGG pathway annotations, which revealed that diverse pathway annotations of these human proteins involved in domain swapping was enriched. So that part also looked into the details. So this is about the application of this model in the human genome. Then we also tried to see the apply these models or this particular random forest model to some plant genomes, and then we choose a few of these plant genomes like Arabidopsis thaliana, and then some tuberculosis, tuberosum, and Lycopossicum genomes. And then we applied the same, we did the same thing, that we downloaded the proteomes, that means the reviewed entries from the UniProt, and then used this model, and we found some similar trend here also, like in Arabidopsis, then 64% of the cases of the proteins were domain swapped, with other plant genomes also showing some similar results. Right. At that time, we were also doing the NGS sequencing of one of the medicinal plant, that is Ocimum, and then we did all the annotation or genome assembly and annotation in the lab. So we thought that we should apply this model to the Ocimum proteome also, and see if what kind of trend we see in this. So the total number of sequences, protein sequences, which were predicted in the Ocimum genome was 36,841, and then around 26%, that is 9,416 proteins were involved in domain swapping. So since Ocimum genome was not highly annotated as compared to the other model of plant genomes, for example, Arabidopsis, so we want to compare this particular results with the Arabidopsis thaliana and see if the trends are same in the Arabidopsis also. So for that reason, we did the orthology check between these two plant genomes by using protein ortho and other tools. A total of 694 protein sequences from Arabidopsis and 941 sequences from Ocimum were used for the analysis. These were the three-dimensional domains were predicted positive protein sequences. We use these two sets of the sequences and compare them in terms of their orthology. Right. So with that, we also look the relationship of these domains of cases in different proteomes. For that, we took the three-dimensional domains for predicted protein sequences of the Tulsi, and we searched against the public database, that is UniProt, and then we try to analyze them, that what kind of similar sequences are present in terms of which are also involved in domain swapping, and then almost 78% of the domains of cases are common to Arabidopsis thaliana, and which shows that the results are somewhere consistent in the different genomes. So these are the results which are obtained when we try to find out the relationship among the orthologs, which are also domain swap cases. Okay. To summarize this part, Tulsi genome has nearly 25% of the protein sequences which are predicted to be involved in domain swapping. Although this is less as compared to the other genomes, the reason may be the annotation or the completeness of the genome. Then the functional annotation of almost 90% of the domain swapped predicted sequences could be assigned. Gene ontology term association was done for almost 85% of the predicted cases in case of Tulsi, and then protein domain association could be observed for 89% of the predicted domain swap cases. Almost 12%, that is 1158 of the domain swap predicted protein sequences are found to be involved in the abiotic stress of the plant, for the plant. So this analysis also we have emphasized that in case of plants, these proteins which are involved in domain swapping have some role in the to cope with the stresses like abiotic stresses in the plants, and also they are involved in the secondary metabolic production pathways. Thank you everyone. And congratulations to ma'am and all of us who are directly or indirectly involved with the lab. Thank you.
Thank you, Atul. It was a great presentation, and I would now like to thank and mention the people who had presented today, and along with your current affiliations. We had Meenakshi, who had presented the dial algorithm in some detail, and also she had explained the domain database very well. And she's currently a research associate affiliated to the Bioinformatics Center of NCBS. And then we had Prashant Shingate, who had presented the structural analysis and sequence preferences at the hinge region, and also pH-dependent structure energetic changes. And Prashant is currently an R&D scientist at the Bayer in Singapore. And then I had talked about how machine learning algorithms had been trained and tested. And subsequently, we had Atul Upadhyay, who had presented its application on genomes. And Atul is currently an assistant professor at the Thapar Institute of Engineering and Technology, Punjab. Okay, so with this, I would like to also request Shafi to present the relevant publications and announce the title for the next talk, next Thursday. Shafi, please.
Hello everyone. So for today, the most cited research we have listed here. We have two early paper for DAIL. One is from Professor Sowdhamini and the next one from Pugalandi et al. And following to that, we have DD-based domain database publication from Vinayagam et al. And then we have a series of domain swapping papers from Shamir and Prashant. This include 3D swap, the 3D swap database and 3D swap web server, also analysis of 3D domain swapping. And for the next week, we have interesting set of talks. We have titled them Structural Analysis of Protein Domains. Please stay tuned.
Great. We had a number of questions which we had attended then and there. They had come in the chat box. So if there are any additional questions, people might do so. You can just raise your hand or so, and then we can invite you to be a panelist. Or you can just put your questions, additional questions on the chat box. Any questions? If not, we have answered many of the questions online. So with this, next week we will meet for, like Shafi mentioned, for the structural analysis of protein domains. So please stay tuned, and yeah, we will meet same time Thursday, 3 p.m. next week. Until then, take care. Bye for now.